Good afternoon, everyone, and welcome to the front row. My name is Jamie Williamson. I'm a professor here at the Scripps Research Institute, and I'll be your host today, where we hear from one of our young stars, Xin Jin. Now, the front row is really our outward-facing lecture series where we try to communicate the exciting work that's going on within Scripps to the community at large. And we've got two groups here. We've got the in-person group. Looks like we've got well over 100 folks that have come to the auditorium. And I know we have over 500 that are scattered around the world, tuning in a virtual format. So welcome to all of you. And we encourage those of you who are local to actually come and come to the auditorium, and we have a little reception afterwards, and you can mingle with Scripps scientists and the speaker and have some great discussions that follow the lecture. So the subject of today's lecture is using genomic techniques to study neurological disorders. So 1 in 5 people are afflicted with some kind of neurological disorder over the course of their lifetime. But neurological disorders are a very complex set of diseases. So some diseases are what we call monogenics, where there's just one gene that causes that, and if you've got that gene, then you're going to have a condition. But what we're finding is that most of the neurological disorders that you might be aware of are really complex sets of genes that can conspire or individually cause some kind of disorder. And one of the reasons for that is that it's all focused in the brain. The brain is a complex tissue. There's all these different regions of the brain, and all of those different regions of the brain are composed of all these different kinds of neuronal cells. They're all organized in some complicated way. So any one gene might be doing various things in different places. So the problem that Xin is tackling is that we basically have this huge list of things that we need to think about to understand what might be called one disease, like Parkinson's. But how do you do that? And it's just a staggering task. So Xin has been here for about 2 years. We hired her roughly almost 2 years ago, and she's an assistant professor in the neurosciences department. And the reason we hired her was that she was instrumental during her postdoctoral studies for developing something called Perturb-seq. And I'll let her explain the details, but this tool is exactly the tool that you need to disentangle the complex genetics of things like neurological disorders. So she has adapted this to study the brain, and what we hope to be able to do is use this to edit genes and address neurological disorders. So I'm going to let Xin introduce herself and tell you how she came to be at Scripps, and she'll give her presentation, and then at the end, I'll join her on the stage, and we'll have a little Q&A. And so if you're here in the audience and you stick your hand up, someone will come to you with a microphone, and we'll try to get you to have a question here in person. If you're in the virtual audience, you can type your question at any time during the lecture into either the chat or the Q&A. And those will get collated, get down to me, and we'll try to get to as many of them as we can. But it's a really fascinating area. Xin's a wonderful lecturer. So I'm going to stop there and introduce Xin. Please welcome her.
Thank you, Jamie, and hello, everyone. It's really a pleasure to be here. I'm very excited to meet all of you virtually and in person. And today, I'm excited to share some of our work introduced by Jamie, really focusing on developing genomic tools to understand the brain. But before going into the science, I would like to spend a few minutes to tell you a little bit about myself and how I ended up being here. This is the photo of a city called Xiamen in the Fujian province in southeast China. This is a city that I was born which you can see is really blessed by the lush greeneries and ocean breeze. And these warm climates make the city Xiamen a really great place for the plant research. And in fact, this is one of the central research sites for the tropical and subtropical plant research. My late grandfather was a plant biologist, and this is a photo of us in front of a research sites in the Botanic Research Institute that he worked on. And many others might be spending their childhood learning, playing pianos or math sessions, and I successfully avoided all of them by devoting myself to nature and with plants. And as a child, you can imagine that very naturally you're attracted I was attracted to my grandfather's research, because all these notebooks of his with this overly sophisticated Latin words and English words just look so mysterious to me, along with these beautiful paintings and drawings of the leaves. And you'll probably know that as a scientist, we're all often overly critical, maybe too critical to our work. My grandfather is a taxonomist, so that means what he studied is how different plants are classified. And many of you know that is actually done by the Linnean taxonomy. And in this kind of classification, oftentimes plants are classified by their morphology, such as how tall the tree is, what is the shape of the leaf, what is the color of the flower. And my grandfather kept telling me that, well, oftentimes for two plants that look seemingly very, very similar to each other, one can be a very precious herb, and the other can be absolutely poisonous. And therefore, morphology is helpful, but it is really not the only thing that helps us to understand the complexity of biological species. So when I was very young, this really planted a seed in my sort of passion for research, and started thinking about how do we actually define biological systems? Is it by their forms or by their functions? And because of these diverse and distinct differences at a chemical and molecular level, is there any ways that we can actually study this almost invisible level of species and understand the biospecies at a level of molecular biology and chemistry? So that's how I started this perhaps a bit unusual path. I attended MIT as an undergrad and obtained my degree in chemistry, where I really want to understand the molecular and chemical level of these complexity in the biological species. And particularly while I was at MIT in 2009, I actually come across the country and came to Ben Cravath's lab and did my summer intern there, which is a wonderful experience. And that was also the very first time that I conducted research as a full-time researcher in the lab. And I absolutely love it. After that, I moved on and completed my degree, PhD degree at Rockefeller University, where I started learning about genetics and started building genetic tools to study the brain, and particularly a small, compact brain of a nematode worm species called C. elegans. And later, at Harvard, I was very fortunate to join a Society of Fellows as a junior fellow, where I had a freedom to be able to bring all these tools and systems and thinking together to integrate chemistry, genetics, and neuroscience. And I found myself very much enjoy connecting and building technology to solve neuroscience questions, which is really an ethos that's been consistent throughout my career. In 2021, like Jamie mentioned, I was very happy to start my lab here at Scripps. And it's very fortunate for me to be here and build this team that many of the lab members are actually here with us today in this audience. I told you earlier that I grew up in the southeast part of China. Before the age of 16, I've never seen snow. And you can see that after almost a decade of snow in Boston and New York City, I'm finally coming back here feeling like coming home and settling. And the early intern back in 2009 in Ben's lab is really an important early imprint that helped me and guide me to come back to this wonderful institute. So in my lab, we primarily build technologies and tools to study brain biology and diseases. and as Jamie introduced, there are many devastating diseases, especially brain disease and disorders, have strong genetic components. In the past decades, the human genetics really telling us what are the loci and variants in our genome are actually carrying out these disease susceptibility as well as resilience. So what our lab has been interested in building and studying can be really summarized in the simple schematics from the left, that we're trying to understand these complex variants and changes in the genome, and how these genome and DNA level gene variants are interpreted and translated across many different cell types. And also at a slightly higher scale, how are these distinct cell types as pieces of a much larger puzzle to put together and organize at the organ level to allow certain brain function, and when the processes goes awry, how the dysfunction and disease can happen. So this is much similar, if you think about it, as my early passion to classify plants and leaves. But now we want to actually understand these individual brain cells. There are many, many different kinds of brain cells, including neuron and non-neuronal cells in our brain, and want to understand their function, and especially their function in interpreting these disease genomic variants. And we want to do so one cell at a time. So I think this is probably not a too controversial thing to say that cells are the very basic and fundamental units of life. Not bigger than that, and not smaller than that. And we already know that our body has many kinds of different tissues, including the skin, the brain, the muscles, and so on. And each of the tissues actually have a village of many different cell types shown here in each of the columns, such as the brain actually contains many different kinds of cells, including brain as well as glia cells. And this diversity of the cell types has been classified by many groups in decades of the work. And now we know that the effort to classify these brain cells as well as other cell types in our body can be done through many different ways, including classify by their location, their morphologies, their molecular composition, as well as many other alternative functions. So now let's put the two things that I've told you earlier together. On the one hand, we know that there are collections of genes, an increasing amount of genomic informations about genes and molecules that give rise to our disease and disorders. And we can perform these genome-wide sequencing, a large-scale comparison, to be able to define at a single variance level what are the changes in our genome that give rise to these resilience or risk to diseases. But on the right side, we also start to appreciate that there are many different cell types, for example, just within our brain. And different cell types have very, very different ways to interpret all these different disease variants that I'm showing you on the left. For example, in the case of the degenerative diseases called Alzheimer's diseases, many of these Alzheimer's disease variants in our genome are actually very specific, residing in an enhancer region of a glia cell called microglia. So you can imagine that there are many different citizens in this village called the brain, and these cell types might have very different kind of interpretation, or you can think about it as tolerance to these changes and variants that lead to disease and disorder. And importantly, these kind of genomics and genetic advancements of this knowledge is not limited to one disease or disorder. There are increasing numbers of many different disorders that has been shown to have strong genetic components, including neuropsychiatric illnesses, neurodevelopmental disorder, degeneration, brain aging, and so on. So it really demands us to start thinking about a platform or a strategy to be able to build together and apply to not just one, but many of these diseases and disorder, and translating these genetic knowledge into actionable biology. So now you can see that this is actually a combination problem. On the left, you have many of these risk genes, and in the case of autism, we'll discuss this later, the number of genes that are contributing to the manifestation of disorder is at the scale of hundreds. And on the right side, you also have many different kinds of cell types in the brain, and each cell type can also interpret with many of these left side risk genes very differently. So it's really a many-to-many problem. So how do we be able to do this rather quickly to not focus on one or two risk genes, but take a systematic approach to take a long list of genes, but also, on the right hand, using high-resolution readout to interpret in each different distinct cell types how are each gene actually playing its role and allow function and dysfunction? We built this technology called in vivo Perturb-seq that allow us to scalably and systematically dissect genome function, but with high cellular resolution. So the development of this technology, Perturb-seq, was really motivated by the intellectual advancement of human genetics. That now we know, for example, in the case of autism spectrum disorder, really thanks to these international cohorts of research collaboration using whole exome sequencing, a form of DNA sequencing technology. And now we have revealed more than 200 risk genes in the year of 2022 compared to what we had 10 years ago. And these are effectively, we call it almost causal risk genes that's contributing to the manifestation of the disorder. So in this plot, what I'm showing you is a list of these almost causal risk genes contributing to autism spectrum disorder and neurodevelopmental delay. On the x-axis, flat here, you are seeing individual risk genes and their names. And on the right, on the y-axis, you can think about the taller the bar is, the higher the statistical significance it is. A lot of these bars are in pink, which indicate that these are actually protein truncating or a loss of function mutation, meaning that this gene is dysfunctional. That's associated with the manifestation of the disorder. So you can see that there are increasingly reproducible genetic variants that's been revealed in the past decades. What we had back in 2012, those 10 genes that at that time has very weak statistical power is in fact the top 10 genes on the left side of these plots, meaning that in the past decade, what we had is more and more empower analysis and the reproducible production and analysis to reveal this list of genetic variants that is consistent across cohorts and across populations. So the question is, amongst these 71 different risk genes, are we really talking about 71 different kinds of disorders? Or is it possible that some of these risk genes actually operate in the same cell type, thus impacting a particular time window of development, or maybe impacting a particular molecular pathway in that cell type, in that time of the development? And it will be important to know to be able to better stratify patients and to understand this very heterogeneous and not so clearly defined disorder. And finally, as I mentioned earlier, we're talking about a lot of these genetic advancement here about autism spectrum disorder, but the similar strategies and the developments are actually also happening in many other disorders and diseases, including schizophrenia and bipolar disorders. So the gene lists are growing, for better or worse, because with this longer and longer gene list, it really demands us to start taking the gene list and start to understand their function and translate them to actionable therapy. In this list of 71 risk genes, some of them have been studied in the past, but majority of them are so-called new genes for geneticists, that there are no cell line made or animal model made. So creating individual model one gene at a time can be very expensive and time costly. And we've been thinking very hard about whether there's a way that we can accelerate this research and build a platform to be able to look at many of these risk genes rather systematically and rapidly. So the birth of Perturb-seq is really the marriage of many different pieces of technology. The first piece, of course, is CRISPR, and is a gene editing technology that was awarded to the Nobel Prize just a few years ago, I think in 2020. And you may already know that just a few weeks ago, the therapy of applying CRISPR in sickle cell diseases was just approved, which is about 10 years from its very initial discoveries in the bench, now in the clinics, which is remarkable. So we took advantage of CRISPR gene editing technology that allow us to go to the genome precisely and programmatically introduce a mutation or change of the genetic variants for a particular risk gene that we're interested in studying. And subsequently, we can actually do these experiments in an animal by introducing these perturbations through viral delivery and taking out these tissues and analyze these cells at a single cell level. So on the right side, what I'm showing you is the analysis of these cells using a technology called single-cell RNA sequencing. So you can imagine that now we can collect tens of thousands of cells and start analyzing their molecular components of each individual cells, and then putting them all together like a Monet painting, that each dot on this plot represents one cell. And the color of the cells usually represents the cell type or the molecular identity. And the closer the two dots are on the plot, it means that the more similar they are in terms of molecular properties. You can think about it as a topographic map of their molecular composition in terms of representation of a very high-dimensional data. So it was established in 2020 when I was a junior fellow that we developed this technology to combine CRISPR perturbation with high-content phenotypic analysis using single-cell RNA sequencing. So how does it really work? On the left, this is a picture of a brain that individual cells, in this case, actually carrying out each individual CRISPR perturbation. And what we can do is to extract and isolate these individual red cells into a tube like this, and then use the single-cell RNA sequencing technology that I told you earlier. And what we end up having is a representation, again, just like the Monet painting. And we can start classifying these cell type into these individual dots so that we can know that later on annotate them by their molecular composition. And you can see that in this particular case, we can classify all the cells we isolated into 5 major groups, including neurons as well as non-neuronal cells at this time of the development. But I want to emphasize that the resolution of perturbations go beyond just these 5 major clusters of cells that I'm showing you on the right. Because what we can do is, in fact, we have the complete molecular composition of every single cell and every single dot on this plot. And what we can do is go in and take each of these 5 major clusters and then further classify them, showing on the right, into subclusters. And as you can see that each of these cardinal cell types are in no way homogeneous. There are many, many distinct villages and different types that we can further classify. And as you can imagine that if the disease risk variance or this mutation impacting only one out of the 5 cardinal cell type on the left, or maybe only one out of these numerous cell type and subtype on the right, we will still be able to capture it and analyze it because we're truly assaying one cell at a time. But if we start blending them and start sort of like using this bulk analysis to analyze it, the manifestation of a rare cell type can be easily overwhelmed. So in this early version of Perturb-seq, we analyzed over 46,000 cells to understand how 35 different risk genes implicated in autism are actually impacting different cell type and their development. What we found is that there are recurrent and convergent cell type as well as molecular pathways that's shared by several risk genes, which is really exciting. And since I started the group here in 2021 at Scripps, we have been really thinking hard about how to make this platform even more powerful and accelerated our understanding. So I will share with you the latest version that we call it massively parallel in vivo Perturb-seq that allow us to actually much more scale up this process of analysis. And this is the work done by Xin Jin. Really is a very talented graduate student who joined my lab as one of the first graduate student cohort. So Xin is a very gifted molecular biologist, and she understands that the limit in this analysis really is our ability to label and harvest enough cells in vivo. So we want to think about a new way to label and harvest cells. And as you can imagine, if we find out a new way to harvest 100 times more cells, Xin can spend only 1% of the effort doing experiments and get the same amount of work done, whereas your students are very smart. So she's really thinking, how can we find a good vector? In this case, she goes to this vector called adeno-associated vectors, viral vectors, which I'll refer to as AAV in the rest of the talk. So this is really a very, very small viral vector that looks like a little hexagon showing here that can pack the gene cargo of your interest. In this particular case, Xin is interested in packaging a guide RNA, which is a key CRISPR reagent to allow you to perform gene editing that she hoped to deliver for the perturbation in the perturb-seq experiment. But how can she find a good AAV suitable for her? What she wants are two things. She wants to have as many of the cells labeled in the brain as possible, but she also wants this cargo, this CRISPR reagent, to be expressed as fast and as high level as possible. So what she did is this very heroic screen that she basically made this library of different AAV vectors. And each AAV vectors can make the cells turn green when they enter the cell successfully, and also express this particular DNA barcode, just like the barcode you have in the grocery, that allow you to decode what item and what AAV this is coming from. So what Xin did is to inject this barcoded mixture of AAVs, and each individually has the capacity to express this green fluorescent protein, and inject them in vivo in the embryo in the developing brain. And she can wait 2 days and ask, what are the cells that turn green? And if the cell turn green, it means that one of these AAV in her original pool has the capacity to enter the cell and make them turn green. And by analyzing the barcode in these green cells, she can come back to her list and find out what are exactly the AAV that was entering the cells. And you can imagine that this is a pool way to analyze many different AAV in one single experiment instead of testing one at a time in the separate experiment. But Xin is not interested in testing only 1 or 2 or 3 AAV. She did a total of 86 different vectors, which is really heroic experiments across many different AAV serotypes listed here that many in the audience might be familiar with or have been using them. But if you have been using AAVs, you will know that typically when we start this experiment, after the AAV injection and administration, typically we wait for weeks or even months to wait until the expression turn on to the prime condition. So when Xin first showed me this experiment and told me that she waited for 2 days and started analyzing the data, I was like, what do I do? First-year student, you have no idea, there's just no way you're gonna get anything. But in this case, I'm very happy that Xin is right and I'm wrong. So this is what the brain section looked like, only 2 days after she injected her pool of these AAV. And you can see that many of the cells in the brain around the dorsal or the ventral ventricles in the developing brain has turned green. And she performed these experiments and analyzed the data, and that helped her to identify a new vector AAV, but now can help her to collect 10 or 20 times more cells and 20 times more efficient than what I could in the earlier version of the technology. So on the right side, long story short, this is Xin-He's perturb-seq experiment that she collected over 50,000 cells that each dots, again, represents individual cells and clustered by their identity. And there are many, many annotations here. But you can appreciate that the greater diversity of different cell types in our brain, but now can be captured in one single experiment. And what's so great about this new AAV that she characterized on the left is that this new technology can allow us to analyze over 30,000 cells within one single experiment. So remember the earlier version of the perturb-seq I have is over 46,000 cells, and that took me over 9 months. Xin did all that within 1 week. So this is really the moments that I realized the sort of incremental improvement of technology can really lead to interesting new biology. So we can look at this dot plot that I showed you in the earlier slides, and if you are like us who stare at these dots for long enough, you start noticing something around the 6 o'clock looks a little funny. So if we zoom in and just pay attention to these orange clusters and orange cells, and they are representing a classes of neurons called the cortico-thalamic neurons. And these are the neurons that reside in deep layer of the neocortex, and they are sending signal from the cortex to thalamus to be able to refine and fine-tuning the sensory input and your perception. It's a very interesting class of neurons in systems neuroscience perspective. But what we found out is that this class of orange cells, the corticothalamic neuron, seems to have a little bit of a population that deviates from its main sort of village clusters. And we found out that this hybrid cell fate that actually was caused by the perturbation of a risk gene, FOXG1, which is a key gene in intellectual disability and autism. So we use machine learning tools to really show that, indeed, this cluster of cells shown here on the left that acquire a hybrid states are actually caused and linked with the perturbation of FOXG1. And the key of this machine learning-based analysis is that we actually didn't set up this experiment to test particularly corticothalamic neurons or any particular hypothesis. We simply performed the experiments in the as unbiased or systematic way as possible and let the data tell us, amongst all the cells we collected, which are the most impacted ones. And eventually, what we can do is not just to appreciate these beautiful dots that I'm labeling here, but what we had is to be able to extract these individual dots, individual cells, and have their molecular components. And here, what we can do is to compare with and without this perturbation, what are the genes that's upregulated, showing on the right, and what are the downregulators. And it's interesting to see that the loss of function of this gene FOXG1 leads to confused cell states, but also the upregulation of many other genes and transcription factors that actually suggest an alternative cell fate choice. And it's interesting to think about normally this gene, when it's present in the cell, its job is to actively repress these alternative choices to make the cells become someone else. But when this gene is lifted and loss of function is happening, these alternative cell fates start to drift away, and these cell populations start to acquire hybrid states. And it's interesting to notice that we actually only observe this phase switch phenotype in this particular corticothalamic neurons, but not in anywhere else in this plot that I'm showing you, suggesting that the action of many different risk genes are cell type and context specific, and again, highlight the importance of using high-resolution technology to read out these effects. So I've been talking about using high-resolution methods, so I thought it's very helpful to sort of visualize why are we really doing it. Imagine the tissue we're all excited about is a Lego brain, and the bulk analysis is to be able to take some pieces of these tissue and analyze them in a mixture, which is still very helpful in giving you strong signals. But the strength of single-cell analysis, as I shown you earlier in Xinhe's screen, is that you will be able to compare apple to apples and orange to orange, and maybe the differences between an Alzheimer's brain and a normal, healthy brain is that the pieces of these blue Lego pieces are a little bit more bluer, or simply you have more of the orangey pieces of Lego, which you can imagine is harder to read out in the left side of the bulk analysis. So Bo-Li Wu joined the lab, and he's not satisfied with the single-cell level Lego analysis. He wants to study the brain as realistic and as similar to the brain itself as possible, and he wants to preserve the spatial location and the anatomy of the brain, which we know are important for its function and dysfunction. So what Bo-Li did was that he took advantage of a technology called tissue clearing. And this is a technology that was developed by Carl Dijssel's and Mark Tesser-Levine's labs. And my colleague here at the neuroscience department, Li Ye, is also a co-inventor of this key technology, that we can take a piece of the brain and make it clear optically that allow clear imaging, but without sectioning or destroy the brain anatomy or the tissue at all. So here, I will show you a movie from Woolley that is coming from a perturbed brain, and each individual cells labeling here are carrying its own different CRISPR reagent and CRISPR loss of function, but now you can visualize where they are across the entire brain without sectioning. It's all transparent and going from the side to the middle of the brain. So we can zoom in and start looking at different brain regions. On the left, this is a brain region called the hippocampus, which is important for learning and memory, and you can appreciate these beautiful morphology and connection of diverse brain cells within the brain region. But the great thing about imaging is that it's like a Google map. You can now zoom out, actually, to appreciate this sort of much more brain region level connectivity. You can see these really beautiful axons traveling, connecting one brain region to the other. And you can also imagine that these really refined and vulnerable axons might be the first target when degeneration or any stress signals or genetic variants come and kick in. And it's really exciting that now we will be able to analyze these cells one cell at a time and also observe their projection changes when the resilience signal or the perturbation signal come in an intact brain. So, finally, I want to summarize what I've told you, and I hope I've convinced you that we are really, really excited about advancement of human genetics to take these genes that are strongly implicated in human diseases, to be able to understand the perturbation of these genes in a causal way will lead to changes of cell states throughout analysis of these cells at high resolution. And this is helpful because it will help us to think about how different variants in the genome can actually give rise to shared or similar or different changes at a cell type level and help us to stratify mutations. And I told you 3 very short stories about our effort in engineering these vectors to achieve massively parallel perturb-seq. And using this technology, we decode a transcription factor called FOXG1 that is a risk gene implicated in intellectual disability and autism. And we identify its very specific role in regulating cell fate, and the loss of function lead to the confused and hybrid cell fates. And finally, I hope you can see that perturb-seq is not just one experiment or one trademark. It can be coupled with many different readouts, such as high-content imaging readout, and build a more holistic picture about how the brain works. And finally, I want to say that genes and molecules, they are very important, but at the same time, we know that genes and molecules don't give us disease or disorders. It is really the nervous system's property. And being able to translate these genes and molecules to define their action sites on cell type, and time window, and brain regions really help us to refine the specificity across these heterogeneous disorders and leading to actionable drug targets and therapeutics. And I think particularly to all the students and trainees in this audience, I want to try to convince you that this is really the perfect time, because now we have all these tools that I talked about, including CRISPR, the single-cell RNA sequencing, tissue clearing, or AAV vectors. These are really the tools that we literally did not have 10 years ago, at least I did not have when I was a graduate student. This is really the right time, both intellectually, the knowledge of genetics, as well as technically. It's really a perfect time for you to join the sort of efforts of functional genomics, and I'm so excited to think about what we're going to discover together in the next 10 years. Finally, the most important slides, we're very fortunate to have awesome collaborators and very diverse expertise. I'm very grateful for all my fundings, especially the collaborative innovation seed fund here at Scripps. It really gave us support, but also confidence as a junior faculty to pursue these high-risk, high-reward projects. And these are the pictures of the heroes and the heroines in the lab who are really doing the real work. There are many exciting projects in the lab today due to the time limit I couldn't tell you about, but they are here, and I'm sure they're very excited to share with you their work. I'm very fortunate to have this group of energetic and brilliant and just fearless young scientists to work with me. They inspire me every day. And finally, Scripps is just such a wonderful place and unique amongst all the institutes that I've worked at. We couldn't have done any of it without the fantastic colleagues, the environment, and the very robust support structure here. Thank you for your attention, and I'm happy to take questions.
That's for you. Yeah. Okay, thanks, Xin. That's terrific. I want to start with a question, and then I've got some here from the virtual audience, and we'll look around from the personal audience. So you had this bar graph, and you pointed out 71 genes, and I've just found myself wondering, how many? So it's the list of genes. It's alphabet soup for most of us. But how many of those genes are expressed in other cell types? How many are specific to the brain? And then when you found those disease-related genes, I mean, I know you were perturbing them, but how many of them really mapped to one particular region in your multidimensional data plot? So was there specificity or not? So that was a long question, but I think you can talk. I'm sure you can talk at length about that.
No, that's a brilliant question. So I think there are certain things we know about this list of genes and these molecules. About half of them encode transcription factor and chromatin modifier. So these are the molecules that interact very intensely with DNA or three-dimensional DNA structures. You can think about how they directly change how your DNA is going to be transcribed, translated, and so on, right? But at the same time, these so-called transcription factors are actually very broadly expressed. They do express in the brain, but also expressed in many other regions. So this is one of the conundrum in biology that just because the gene expressed doesn't always say, well, it's important there, or it's important for this particular moment, right? So we think it would be important and really interesting to take a perturbation approach, which is to take the gene away to see whether the cell is still functioning. It's like when you want to know a particular parts of your car is important, just take it away. This is the genetics sort of analogy of thinking about what is really the function using a perturbation approach. And we're very excited to figure it out.
Terrific. So we have a question from the audience down here. Oh, I thought we did. Oh, yes.
I have a question about I have a question about the specific perturbations that you introduce. Surely they could have a different effect on all of those different genes that you're looking at. How do you account for the different effect that it can have on the genes and compensate for that variability on the output when measuring how much the cells were upregulated or downregulated when you're looking at those beautiful plots at the end?
Can I ask for the clarification of the question? How do I account for the variability of?
Well, let's just say some perturbation introduced a gain of function in one gene. Are you certain that that same perturbation would have the same effect on all of the genes you're studying at the same level? And if not, how do you account for that at the back end of the analysis?
So a particular perturbation that we can extract it from this particular cell, right? And then what we are doing is actually compare to a control cell that has everything else and also some control changes, but not about this gene. So I feel like it's a comparison not with cells outside and never experience this, but rather internally experience everything else similar to this perturbed cell, but minus that particular perturbation, if that's your question.
So every cell that you're looking at, you're comparing it to a control cell that's kind of a mirror of what it would be without the perturbation, rather than all of the other cells in the sample?
Yeah, that's exactly right. So we always ask perturbation effect with the control group and within the same experiment. So it's like an internally negatively controlled experiment. And especially, we don't really expect to see a lot of similarities, right? So you can also think about these 71 perturbations, they're kind of controlled for each other. Mm-hmm. And another thing I should mention that we're not just building statistics on one or two cells, right? Like you will be able to consistently have groups of things and you can actually regress against all these other confounds and think about how significant was the effect sizes. Yeah.
Awesome, thank you.
Thank you.
Xin, we have a couple of related questions that I'll aggregate from the virtual audience. But you talked at the end about using gene editing to actually treat disease, and so that's the first one has just been approved. So, talk us through the arc of what that would look like. You would find, okay, I can repair this gene. How would you go from that observation and beyond your lab into the clinic and actually a treatment strategy? So just, I think people are generally interested as, I think we see where you're starting, but how do you get there, and what's the arc of that look like?
Yeah, no, that's a great question. So we're talking about hundreds of genes that give rise to disorder, right? And you can imagine, sure, maybe we do hundreds of different treatments, and that's one way to think about it. But I would say it will be interesting to even figure out amongst these hundreds of different changes in the genome, maybe 5 of them, although they are different genes and different mutations, what they are doing is actually one thing in this particular classes of neuron at this particular time, in this particular brain region that we need to reverse. So we're not really targeting the primary sort of mutation, very similar to sort of cancer drugs, right? You don't really target a very primary mutation, but you know that what's the immediate next thing that is very important to reverse. And the beauty of it is that then we're not dealing with, hopefully, 100 different treatment, right? Individually need to have FDA approval and toxicity and all that. What we can start thinking about is maybe this is obviously not one disorder, but hopefully not a 100 different kind of disorder. The number is between 1 to 100. But if we can group, let's say, 10% of these mutation or changes in our genome into a subtype that help us to stratify and start understanding when and what and in what cell type and what specificity is sufficient to reverse at the molecular level as a start, I think that would be a very, very exciting way thinking forward.
Right, okay, so you're not necessarily going to be targeting these disease-associated genes, but perhaps there's 10 or 20 of them that all point to some common point, and then you can adjust the expression of that gene to compensate for 10 different possible defects.
Absolutely,
That's
absolutely.
sort of what you're saying.
Yeah.
Okay, great. Another question from the virtual audience. So one of the things you're doing is you're generating a lot of data, and so that's fertile ground for the application of machine learning and artificial intelligence. And so a couple people asked about, how might you use that, and can that expand the insights that you get from these enormous data sets that you're generating?
Yeah, I think it's one of those things that you start by being able to produce a lot of data, and you're like, oh, but how do I analyze it? But then at the same time, you push forward, and the experimental ability to generate better and more scalable data always sort of like a year or two precedes your ability to really sophisticate it. So we're still thinking about this is active discussion in lab all the time. On the one hand, I would say we can always ask specific questions. So just because you have a lot of data doesn't mean you cannot ask very specific hypotheses. So we're still doing that. But at the same time, we'll start exploring what is the data structure out there. So you can think about these single cell and large scale analysis. It's like a huge matrix, right? Let's say you have 1 million cells, and each cell has 20,000 genes, so it's a 1 million by 20,000 genes, right? But it's also important to be not too overly paralyzed by this matrix, because most of the matrix are zeros. Right? It's an extremely empty matrix. So there are this kind of like so-called sparsity of the data is what's similar to a lot of these machine learning tools are very good at handling, such as the large language model that we're all very excited about, ChatGPT. It's actually also the same challenge that data is huge, but it's also very sparse. So we're actively thinking about adopting these machine learning tools to be able to sort of perform analysis, but also we know what we're looking for, we know what's changed, and we ask the machine learning tool to do its own sort of like a million monkey on the typewriter, being like, oh, can you see also what we expect to see using the more sort of hypothesis-driven approaches? And I think this is the phase that we start testing both grounds. It will be really exciting to see what we can actually build. Now we can apply them to these increasing large data sets.
So can you use gene therapy to treat neurological disorders, all of the ones that we're all aware of, Alzheimer's, Parkinson's. Is there any prospect for that kind of therapy? And is there any way that your basic research would inform how to do that?
Yeah, so one of probably the most prominent or sort of like the most frontier example will be the Rett syndrome, which is caused by a single gene mutation, MECP2. In this case, it's very similar to the examples I was talking about. It's a loss of function and lose a single copy of this gene. So it's been a lot of sort of clinical sort of exercise on thinking about how to apply gene therapy for Rett syndrome. So if anything, I would say this is the very much frontier of a single gene disorder. But it's also very challenging for several reasons. First is the delivery, to be able to effectively deliver this reagent to the brain. And the question is, when do I deliver it? How many cells do I have to deliver to sufficiently restore the gene function to be able to restore at a sort of patient symptom level? And then some of these genes are also very sensitive to the dosage. You cannot give it too much, cannot give it too little. So there are a lot of the molecular therapy and development has been happening. But it's a very, very good question. And I think last time I talked to these Rett syndrome clinical developers, it looks like they're getting very close. So maybe we'll hear something from this frontier, hopefully in a few years.
So it's really staggering to watch the progress. You talked about in 10 years ago, there's 10 genes, and now, and you really made a great case for developing technology that lets you get more information. And so there's just this avalanche, but so 5 years is a huge amount of time, if in terms of the amount of evolution that's gone on in this field. So where do you see the next 5 years? So where are you going to be in 5 years? Where do you hope you're going to be?
Yeah,
Big picture.
it's a great question. I like to think, things are always going to get better, faster, more scalable, right? But I also like to think there's an inflection point that before a certain point it's kind of going linearly, but after a certain point it just explodes, right? And that's the point that I feel like is the most important for any researcher or young investigator to start paying attention to, right? And I think for genomics, it's pretty clear, and technology like this molecular tools that we didn't have before. And I think the inflection is probably around this time. And in the next 5 years, I think we will have a pretty decent chance to be able to at least perform these experiments and ask whether there is a convergence amongst these 71 genes. It's probably not all 71 genes do one thing, because if that's the case, we would have figured it out. We mean collectively the community. But I think that's something that we can very concretely solve.
Okay, well, I think we're coming up on the end of our time, so I look forward to hearing more about the exciting things you're going to do, and I'm just going to watch with wonder. So we're at the end of our calendar year for the front rows. This is the last one for 2023. There is one that's I'm sorry, I think I forgot the date, January 10th, I believe. And it's our president, Pete Schultz, who's going to talk about, did I get it right? Yes. Okay. I have all my genes.
Excellent genes.
But Pete's going to be giving kind of an overview of what's going on for drug discovery at Scripps. So we hope that you will all come in person and join us outside for a reception. So for those of you who are here, please come out. We'll talk some more. For those of you who are virtual, thanks for joining us. This is all going to be available on the web, so you can tell friends. Go to the Front Row webpage. And as for January 10th, you will find me in the front row.